Okay, the next three slides are not slides that I'm going to test on, and so you can look at them. It's there. I'll only mention one or two things that I think are of general interest here, but this is not a pathway you have to memorize because we got enough of those, don't we? So mTOR is going to be one of these powerful molecules involved in particular, T cell activation. And there is a drug that is used to block T cell activation, which is called rapamycin. But if you work in a hospital or somewhere around healthcare, you may see this called sirolimus. That's the other name for this drug. Zerolimus. Well, close. Sirolimus. Anyway, these things are given to prevent organ rejection, and stents are actually coated in this also to prevent sort of buildup of inflammatory factors in the body. And so there's a whole pathway. Oh my gosh, there is. And so this is where AKT is going to come in. You can see it's going to bind to a PIP3 molecule, use that as a little anchor, and then we're going to activate, and eventually get mTOR activated. So that's fine. And then the other side is going to actually use this same complex that we've mentioned before. So we got our LAT here with all the phosphates on it. We got our SLP76, and we got our GADs. And these proteins, remember, were phosphorylated by ZAP70 way back when, several slides ago. And so what this is all going to do is that we've got another one of these little G proteins that's going to be bound to GDP. It's going to open up, pick up a GTP, and by doing that, it is going to dock with the tail of these proteins, and it's going to cause the head to change its conformation so that it can now be more active and do other things. And there are a bunch of other proteins involved in this complex that each, one activates the next. That's how all of cell biology is. Okay, so what about CDC42? CDC42 is going to be one of the factors that causes cells to just become very active, and it's going to push them through the cell cycle. It's going to be associated with cell division. So how do we activate this thing, this little CDC42? Well, CDC42 is a G protein. So it's going to normally be holding on to a GDP until it picks up a GTP. So it's a little GTPase. All right, so we've got our PIP3 molecule up here, serving as an anchor, and a wonderful anchor for VAV. Yeah, like vavoom. VAV is then going to bind to phosphorylated SLP76, which is of course attached to GADs and LAT, because that's what those three proteins do. Down here at the bottom, this is our protein called WASP, which is associated with the disease right back here, Weisskopf-Aldrich syndrome. WASP is in an inactive state on its own, but it's going to interact with NIC, which interacts with phosphates on the tyrosine, on SLP76. So, when NIC has interacted with GADs, or with SLP76 phosphotyrosine, it causes WASP to open up. This allows CDC42 to bind and become activated, and activated CDC42 will then bind to WIP. And WIP will actually cause actin to directly polymerize. So this is actually going to directly make the cytoskeleton start to grow in a particular way. And so this is going to be associated with cell movement and cell activation. Remember, the cells are going to change shape when they're activated versus when they're not. They tend to be kind of rounder and less actin when they are at rest. So, how does this work? Well, this is going to be a thing that happens at the immune synapse. So here we've got a T cell, and since it's got CD4 on it, we know it's going to be a helper T cell. And so there's our T cell receptor, alpha and beta. Here are our CD3 molecules, gamma delta and gamma, no, gamma epsilon and delta epsilon. There we go. Here are our zeta chains, and remember there are ITAMs on each of these and 3 ITAMs on each zeta chain. Remember that attached to the bottom of CD4 is going to be a LUC kinase, which when CD45 activates it, the LUC kinase is going to put phosphates onto all of these little ITAMs. Remember that ZAP70 is going to bind to the ITAMs with their little phosphates, and then it's going to go off and activate other things like we'll put phosphates on LAT, which will then bind to the other proteins, I think, and eventually SLP76 GADS. There, that's the one I was fishing for. I was thinking, is it GADS? No, I didn't want to say, but it is GADS. That's fine. Yeah, and so all this is going to happen at a little area called the immunological synapse, where multiple T cell receptors can make contact with multiple MHC molecules. And so in this case, it's going to be an MHC class 2, but the immunological synapse would look roughly the same if it was MHC class 1. It's just that there would be a CD8 protein leaning over and interacting there rather than a CD4 protein. Everything else happens the same, just makes it a different kind of cell. And so the purpose of all this is that once we have a connection here, and we phosphorylate everything, and we make these signaling complexes, we activate phospholipase C, we are then going to cause some changes at a transcriptional level. We're going to make new genes. We're going to translate them into proteins, and then right in the middle of these things, we're going to secrete some of these new proteins that we've made. Those are going to be the signal that tells the cell that is presenting the peptide that yes, you have found something, or that you are very infected and need to die. Yeah, depending on whether we're talking about class 1 or class 2 presentation. Okay, that's the end of T cell signaling. Isn't that nice? Let's actually pause here, then we'll go through a bit of B cell receptor signaling, which is really not as bad, I think, or as detailed as T cell receptor signaling. And then, we'll finish out this chapter. This is going to be the last section of Chapter 7. And Chapter 7 is the last chapter we're going to cover in this course. So, oh my goodness, we're almost done. Thanks very much.